In the United States, pediatric cancer survival rates have climbed to 87%, and the FDA has approved 156 distinct immuno-oncology therapies. This split screen shows a sharp divergence. On the left, you see the rapid accumulation of those 156 domestic FDA approvals over the last 14 years. On the right, global breast cancer incidence is projected to surge, climbing past 3.2 million new cases annually by 2050, with mortality rates in lower-income nations remaining largely stagnant. We have engineered highly specific biological weapons against tumor cells, but the clinical deployment of those weapons is failing to intercept the geographic reality of where patients are actually dying. To map the current state of oncology, we have to look past the top-line approval numbers and deconstruct the landscape into three distinct layers. The first layer is the biology itself, the extreme targeted precision of our newest molecular engines at the cellular level. The second layer is the economic pipeline, the commercial saturation and institutional risk aversion that dictates which drugs actually get funded. And the third layer is the epidemiological reality, the systemic global infrastructure required to diagnose and deliver care. Relying strictly on a high domestic FDA approval count to measure progress creates a dangerous illusion of victory. It ignores the severe systemic bottlenecks keeping these drugs from the patients who need them most. At the very top of the biological sphere, precision medicine is working flawlessly for highly specific genetic subgroups. Consider patients with mismatch repair deficient, or dMMR, solid tumors. These tumors have a broken DNA repair mechanism, causing them to accumulate thousands of mutations. When immune checkpoint blockade therapies strip away that camouflage, the patient's T cells suddenly recognize and systematically destroy the heavily mutated cancer cells. The clinical outcomes for this specific patient subgroup are staggering. Recent trials have demonstrated complete clinical responses using immunotherapy alone. The tumors vanish. By achieving these complete responses, roughly 3% of patients with dMMR solid tumors could potentially avoid radical surgery, radiation, and chemotherapy altogether, overturning established frameworks for curative intent care. This extreme biological targeting is also accelerating within pediatric oncology. Comprehensive sequencing has revealed that more than 70% of pediatric tumors harbor clinically actionable genomic alterations. These insights are clearing regulatory hurdles today. The FDA's recent approval of the targeted therapy tovorafenib, specifically for BRAF-altered low-grade gliomas, is a direct result of profiling and attacking exact molecular vulnerabilities. Adult malignancies typically evolve over decades from environmental exposure or age-related mutations. Pediatric cancers originate in immature cell types and are driven by deep disruptions to normal developmental programming. Decades ago, pediatric cancer care relied on the blunt application of chemotherapy and radiation in clinical wards like this. We are now isolating the exact chromosomal rearrangements and epigenetic alterations fueling the disease. At the purely biological level, we have successfully engineered interventions that achieve curative intent efficacy for genetically defined populations. Yet, if you zoom out from these precise victories and look at the macro development pipeline, the picture changes entirely. Of the 156 cancer immunotherapy approvals through 2025, 120 are immune checkpoint inhibitors. Within that, saturation is extreme. 115 rely on a single molecular axis, the PD-1/PD-L1 pathways, leaving a tiny fraction for novel biological targets. This narrow focus is a reaction to high-profile clinical failures. When developers tried to target the next generation of checkpoints, like TIGIT inhibitors, the results consistently fell short in late-stage trials. These failures exposed a severe gap in our testing infrastructure. The preclinical animal models we rely on have proven highly unreliable at predicting how complex human immune responses will actually behave in a clinical setting. When biological risk increases, economic behavior tightens. The fierce competition for institutional funding dictates exactly where research capital flows. This creates perverse incentives for academic researchers and pharmaceutical sponsors. Securing hundreds of millions of dollars for a phase 3 trial demands high certainty. Rather than pursuing untested biological axes, sponsors pour capital into incremental advancements on validated targets. It is financially safer to develop the market's 10th PD-1 inhibitor than to risk capital on a novel mechanism that might fail. The FDA recognized this gridlock and initiated a severe regulatory shift in 2025. They forced pharmaceutical sponsors to convert 6 accelerated approvals into regular status by providing hard evidence of clinical benefit. Empowered by recent legislative reforms, regulators are effectively ending the era of perpetual accelerated purgatory, where duplicate drugs could sit on the market for years without proving they extend patient survival. The high volume of FDA approvals tracks with commercial saturation and a risk-averse funding ecosystem, leaving broad biological innovation as the outlier. If the domestic pipeline is economically bottlenecked, the global reality is constrained by pure physical infrastructure. A nation's Human Development Index, or HDI, a composite metric of life expectancy, education, and income, is now the primary determinant of modern cancer survival. For female breast cancer, latest data indicates 1 in 20 women worldwide will be diagnosed in their lifetime. This map demonstrates the stark divide in outcomes. High HDI nations see the vast majority of diagnoses in older populations. In Africa, however, nearly half of all breast cancer cases, 47%, are diagnosed in women younger than 50. Severe delays in diagnosis, treatment abandonment, and a lack of basic oncology infrastructure drive this disproportionate mortality and younger onset. We see this exact same infrastructural stratification across pediatric oncology. Roughly 90% of the 400,000 children diagnosed with cancer each year live in low and middle-income countries. In those regions, the 87% survival rate we see in the US plummets to below 30%. At the macro level, a patient's geographic location dictates their probability of survival far more than the invention of a new immune checkpoint inhibitor. But you don't have to look at global epidemiology to find infrastructure dictating access. It is happening in domestic policy, hidden within the economics of drug formulation. Consider the recent wave of subcutaneous immunotherapy approvals, like the new injectable formulation of pembrolizumab, the world's top-selling oncology drug. These subcutaneous shots reduce a 30-minute intravenous infusion down to a 2-minute injection. Publicly, this is framed as an equity intervention, a way to reduce clinic burden and potentially allow for home administration. Underneath that logistical benefit lies a highly effective commercial strategy known as patent evergreening. This timeline tracks the patent lifespan. The primary patent for the original IV formulation of pembrolizumab expires in the US in 2028. By shifting clinical practice to the new subcutaneous formulation now, the manufacturer secures a new exclusivity window extending years into the future. Many domestic updates celebrated as access improvements function as commercial maneuvers to protect market share, successfully delaying the entry of lower-cost biosimilar drugs, artificially sustaining high systemic healthcare costs while complicating the economics of true health equity. To measure oncology in 2026, we plot 3 layers: biological efficacy, pipeline diversity, and global penetrance onto a matrix. The data clusters awkwardly. We score high on targeted efficacy, poorly on pipeline diversity, and our global penetrance is severely bifurcated by geography and income. With 156 approvals and complete clinical responses in dMMR tumors, the core biological principle of immune-based treatment is a proven clinical reality. But the next era of oncology cannot be achieved by safely funding the market's 11th PD-1 inhibitor. When early trials show response rates exceeding 50%, randomizing patients to known inferior treatments becomes an ethical liability. We need to formalize the use of synthetic control arms and crossover designs to move effective drugs to market without exposing patients to suboptimal care. We must restructure institutional funding to incentivize high-risk research and simultaneously build the international infrastructure required to deliver these complex assets to lower-income populations. Regulatory approval is merely a milestone. In modern medicine, global clinical penetrance is the only outcome that actually saves lives.